Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello students, welcome to the TV learning series. As you know, we're going to be studying biology lesson today. I'm teacher Herbert again, who will be taking you through the biology lesson of today. The biology lesson of today is concerning about practical 1, which is concerning about food testing or food test. We're going to have a number of food tests of a number of food compounds, that we're going to be testing today, and we try to make an observation to see what are the color changes, and we make a conclusion of saying, or a deduction of saying that the food contains a particular kind of nutrients. The food we are going to be testing today, we are going to be testing starch, we are going to be testing proteins, reducing sugars. If I say reducing sugars, I mean examples of reducing sugars. We have glucose, we have galactose, we have fructose. But the example we're going to be using today, we're going to be testing glucose as an example of reducing sugars. The next, we're going to test about non-reducing sugars. If I say non-reducing sugars, I mean the sucrose as an example of non-reducing sugars. We are going to be testing fats. We have a number of foods that contains fats, and we're going to be testing some of the foods and determine whether it contains fats. We're going to use also to test vitamin C, or what you call in biology, it is called ascorbic acid. Student, you're also going to be using different chemicals or reagents that we're going to be used to test those food nutrients. Here are the chemicals that we're going to use. Chemicals that are going to be used. We're going to use, number one, the iodine solution, or we call it to be iodine reagent. Number two, we're going to use Benedict solution, or reagent. Three, we're going to use burette reagent. We have number four, we're going to be using ethanol. Number five, here on burette, we can use burette, we can also use Million's reagent. Or you can also say, or you can use copper 2 sulfate. As you know, the chemical formula of copper 2 sulfate is written like that. That is the chemical formula. We're going to use ethanol. Number five, we're going to use DCP-IP. In full, we call it to be endrophenyl dichlorphenyl. Endrophenyl dichlorphenyl, which is DCP-IP. Yes, sir. Hey, that are, let me say chemicals. Chemicals stroke reagents to be used.